microphone. If you can hear me, please let's start the lesson. Can you hear me, please?
Yes.
Oh. Janina? Yes, Janina. Jemima and Soro, can you hear me? Angela?
Yes, teacher.
Can you speak? I can't hear you people.
I can hear you.
Okay, yes. Someone said yes. All right. We are looking at our topic, nutrition living organisms. And we know before we broke off, we had really looked at some of this work, but just a recap of what you saw. Nutrition 1, it is what we call feeding, and there are two main types of nutrition. That is the autotrophic nutrition and heterotrophic nutrition. Can someone tell me what autotrophic means? Yes, Katrina. What does autotrophic mean?
The world
nutrition, what does it mean?
with passcode. The world 21 season.
Asoro? Jemima?
Yes, teacher.
Hello?
Yes, teacher.
So autotrophic is about self-feeding. There are some organisms can make their own food, whereas heterotrophic means different feeding, but some organisms get food from other sources. Okay, so for the green plants, they are able to make their own food. Some bacteria can make their own food. We call them the chemosynthetic feeders, or chemosynthetic nutrition. Then, if you look under heterotrophic nutrition, we say it can be grouped into okay. If you can hear me, that's good. Okay. Into saprophytic nutrition, parasitic nutrition, parasitic nutrition, insectivorous, epiphytes. This is what we looked at before. However, if you can hear me, I need a response from you. If you ask a question, please unmute and then give me what? Give me a response. All right. Now, for depending on the mode of nutrition, the intake of food, nutrition can be grouped as saprophytic nutrition. You know about saprophytes. You know about parasites, holozoic nutrition. We feed holozoically, as humans, yeah. Of course, insectivorous feeding. We have some organisms that feed on insects to get nutrients, like the leaf pitcher. You've heard about. There are some plants live in nitrogen deficient areas, and they need to get nutrients. So they capture insects and feed on them. Those are insectivorous insects. So it is insectivorous feeding. When we have epiphytes, we would have talked about some plants that get support from others for purposes of feeding. We find this in the forest. Epiphytes are not actually parasites, but maybe they get support when they live, like in forests where plants have larger canopies, so they can't grow down. They need to get attachment and also compete for light. For that case, okay. Then this is what we looked at before. Then those are details of symbiotic feeding, predators, If you talk about predators, these are carnivores which chase and kill their prey to eat. Like scavengers are those. There's a difference between a predator and then a scavenger. Predator will kill its prey, like a tiger, a cat. A cat can't find a dead rat and eat it. No, it must kill the rat and then eat it. That's what makes it a predator. But a scavenger can find anything dead and will eat it. Yeah, jackals, hyenas, those are not predators. They are scavengers. Yeah, in real terms. Okay, they feed on meat, but those which has been killed by other organisms. All right. The food we eat, must have components, must be rich in energy, food we eat must be bodybuilding, food we eat must be protective. So all the food we eat, we aim at those three things. Then that is what we looked at. Then about what we saw about autotrophic nutrition, that is photosynthesis. Okay. Can someone define for me photosynthesis from the screen I'm sharing? Yes, Innocent. Can you read for us? Look at your screen and then read. Kindly look at your screen and then read what is there for us. Okay, Kaitesi? Yes, Kaitesi. Yes, I can see Kaitesi, yes. Go on. Yanina or ITEL 23, go on, please. Read on the shared screen. Yes, Kaitesi, I've chosen you. Yes, Kaitesi.
Yes, I'm
Good.
speaking.
Kaitesi?
Yes. Fiona.
Hello? Can someone read for us the definition for photosynthesis? Someone read for us the definition for photosynthesis. Look at your screen.
Photosynthesis
Any participant
is a person.
Hello?
Yeah.
Hello?
Yes, teacher.
Yes, Nasoro. Yes, Nasoro. I can't hear you. Yes, Nasoro. Okay, Kaitesi.
Photosynthesis is a process by which green plants, having chlorophyll, synthesis, simple sugar.
Hope you can hear me. Anyone who can read for us? Peter? Katrina? Marcus? Kayanja? Innocent?
Photosynthesis is the process by which green plants having chlorophyll synthesize simple sugars, glucose from simple inorganic
you can hear
materials.
Clearly, I need to hear from you. This is an interactive lesson, so I need your response from what I'm talking about. I've shared a screen, and I want us to do a recap of what we did before. Okay, so that's why I need someone to read for me what I've shared on your screen, if you are following.
Photosynthesis is the process by which green plants have...
Okay. Let me continue. Okay. Okay, someone tells me there's a problem with my speaker. Okay. Hello. Hello.
Hello?
Hi, teacher.
Yes, Kaites, now I can hear you.
Okay, so I'm
Yeah.
reading
Okay.
photosynthesis is a process by which green plants having chlorophyll synthesize simple sugar in brackets from simple inorganic raw materials like
Okay.
carbohydrate using energy of the sun.
All right. Okay. So it means that by this process of photosynthesis, green plants use their chlorophyll and they make sugars or glucose from raw materials like carbon dioxide, then water. These are simple materials, okay? And then they need energy, which energy comes from the sun. The byproduct for this process is the oxygen, which is released in the process. And down there we have an equation for this process that we have carbon dioxide plus water in presence of sunlight, which is trapped by chlorophyll, shall produce glucose, shall produce water, and then the oxygen will be given out. And this sugar which is produced is converted into starch, which is now kept in plants in the form of glycogen. And these are the food results that one can be used by the organism itself, the plant, or other organisms which feed on the plant can use the same when it gets into their living cells. When we eat these carbohydrates, that is what we get. You remember, we are looking at going through what you looked at before. The site of photosynthesis, we agreed that photosynthesis occurs in the green parts of the plant, any green part of the plant, but most leaves. Although if the stem is green, also photosynthesis can take place. If the fruit is green, some photosynthesis can take place because there is a chlorophyll. Green shows there is a chlorophyll. However, you need to understand it is not only chlorophyll which is required for photosynthesis. Plants have other pigments. Even plants with other kind of pigments, we can carry out photosynthesis. Not only the green plants do carry out photosynthesis. Even we have bacteria which manufacture their own food. We call them the chemosynthetic bacteria. Even so, the issue here is that there are very many different types of photosynthetic pigments, or pigments which can be used for photosynthesis. But the most common to us is the one we call chlorophyll, which is green. Even chlorophyll has good types, but we shall not go into those details. So for photosynthesis to take place, we remember the requirements were now on the requirements for photosynthesis. We have chlorophyll. We have seen the importance of chlorophyll. Then we have carbon dioxide, which we see as a raw material, and which is abundantly got from the atmosphere. We're on page 5 if you have finished writing these notes. Then the other requirement is water. The water is abundantly available, okay, unless the plant is living under conditions of water stress, like in drought. But there's a way plants deal with that. So then sunlight, which is also plenty available during the day, meaning that photosynthesis can take place during the day. Then also talk about why should plants have photosynthesis, the importance of photosynthesis. One, providing food, okay, then getting oxygen for respiration. Many organisms need to survive because of respiration, and how they do this by using the oxygen which is produced during what? Photosynthesis. We might use plants very much because they carry out photosynthesis and we get the oxygen they produce, and we also rely on it. However, we also looked at the factors which can affect photosynthesis, and these are 3 mainly. Can someone read for me those factors? Those factors which affect the rate of photosynthesis? Yes. Factors.
Light.
Okay.
Temperature.
Very good.
Carbon dioxide.
Very good. Now, these are the factors we call the limiting factors Okay, these factors are called limiting factors to the process of what? Photosynthesis. If you remember in class, we may have talked about what we call a limiting factor. Yeah, and say in a process, for example, photosynthesis, which is affected by more than one factor, at a certain time t, okay, there is a factor which is closest to the minimum. So at any point, if there are three factors in such a situation, the factor which we call a limiting factor will be the one that will be lowest in supply. And if we want to increase the rate of the reaction or that process, then we have to increase or decrease that particular factor. Okay, so without changing the other one, for example, like photosynthesis, if there are three, if we have too much light, we have enough carbon dioxide, but again there is no light. Okay, it means that now light will be the limiting what? The limiting factor. I normally explain limiting factor in the way like if you are at school, teachers are there, they have provided food, lessons are going on, but again you don't have pocket money. Pocket money is very important because one, what happens during break? What happens when books get finished? You know, so your studies may be affected by a certain factor which we have to look at. So if you're looking at your poor performance, we have to look at many things. There could be a limiting factor, which could be like maybe pocketing money. They send you to school, that's for pocketing money. So your academic progress is affected by that factor, which is under limited what? Supply, and this time it will be pocket what? Pocketing money. If they give you the money, maybe pocketing money, and they have not paid the fees, you'll be chased away from school. Because so school fees will be the limiting factor. As you're reading, you think about, oh, they have not paid fees. You're chasing, you know, that one can be a limiting what? Factor. So if a process has many factors affecting it, always there will be one which is under lowest supply that can affect its progress. That is what we call the limiting what? Factor. And we yes, please. Yes, please. Yes, Katesi or Bridget, someone talking?
No, it's not me.
Okay. If you're not talking, then put off your microphone, eh?
Okay.
Yeah. Okay. Anything so far? Do you have anything we can hear from anyone so far? Any challenge? Can we continue?
Yes.
All right. So we had reached at the limiting factors. I hope you have this work written down, or you had even written before. We are now on page 7. There we are describing, for example, how light intensity can increase the rate of photosynthesis, and you know light intensity can vary. Yeah, you can have low, moderate, or very high light intensity. But if you increase light intensity, like in this case, when you look at this graph, a point comes when the rate becomes constant. That means now, even if you have provided light, it is no longer light you need to increase that rate of photosynthesis. There should be another factor. Maybe this time it could be carbon dioxide concentration or another factor apart from what? Light. Out of the 3 factors which affect rate of what? Photosynthesis, which we have seen. And those factors, if you remember, saw them as temperature, light, and then carbon what? Carbon dioxide. So if you look at your graph and it's becoming flat, yeah, it means that factor you're trying to increase, like in this case, we're increasing light intensity. So it means that light intensity, it will only cause an increase up to a certain limit. When you increase and the rate remains the same, or remains constant, like in that curve, it means that now there is another factor which is not light. You have provided maximum light, but maybe temperature needs also to be raised. If this rate is what? To increase. That is it. Also, if you remember, we talked about oxygen, that oxygen can reduce rate of photosynthesis. That is, if you increase oxygen up to 30%, it can affect the rate of what? Photosynthesis. Now, these are other factors which can affect photosynthesis, like mineral elements, pollutant, chlorophyll content. However, you just remember we have only 3 limiting factors, but there are other factors in play which can affect the rate of what? Photosynthesis. Now, this one also takes me to other factors like number of stomata, Some plants can have few stomata, others can have more stomata. If the stomata are many, which are open, it means that there is a good exchange, gas exchange to occur, and then the rate will be high. If stomata are closed, it means that rate of photosynthesis will come down because we need materials from the environment, that is from number of stomata. And then surface area of the leaf, some leaves are too small, others are large, okay? That's why you find some leaves are broad, others are like spines, are too small. So it means that the size of the leaf, okay, or the surface area of the leaf can affect the type of what? I mean, the rate of photosynthesis, of course. Now, this one now brings me to the factor that is experiments on photosynthesis, and particularly testing a leaf for starch. This lesson is for all senior 2. In one way or the other, you may have looked at this work, and some groups may not have looked at it. That's why I'm going through this work so that we can be at the same level at a particular time. Okay? So we are going to look at how to test a leaf for starch. Can someone read for me the materials needed for this experiment? Bridget, Sachide, Rooney. I'm calling on someone to read for me the materials for the experiment we are going to discuss.
Green leaf.
Okay. Yes, Bridget.
Green leaf.
Okay, very good.
Green test tube.
Okay. Okay. I've had those two. Katrina, can you take on? Yeah. The third one, Katrina.
Ethanol.
Ethanol, very good. Okay. Water bath, okay. Very good. Very good.
What iodine solution?
Yeah.
White file.
Actually, it is a white tile. That was a typing error, okay? You can correct that in your books, eh? May I know, do you have your books with these notes, eh?
Yes.
Yes, so you can correct that one. It is a tile. You know tiles, eh? Tiles,
Okay.
eh? Yeah, it is supposed to be a white. It's supposed to be a white tile. Okay. Now, like you said, the green leaf. Now, this green leaf, we need to get it from a photosynthesizing plant. And where can we get a photosynthesizing plant? Where can we get a photosynthesizing plant? I test it. Yes, Kaitesi. Yes,
Yes, teacher, I beg your pardon.
I said for this particular experiment, we need a green leaf, right? Which has been photosynthesizing, all right? If I am looking for a photosynthesizing leaf, where do I pick it from? Where do I get it?
If you're looking for a leaf.
Yeah, which is a green, which has been photosynthesizing. Where do I get it?
From a tree, a leaf.
Okay, so I'll get from a plant that has been exposed to the sun. Okay? I'll pick a leaf that from a plant which was exposed to the sun for some period of what? For at least 2 hours, let's say. Okay? Yeah, so that's what I would need. Then I would need a test tube for boiling, because it needs boiling. So you either use a test tube somewhere or shall use a boiling tube. Then methylated spirit, we are going to see where it is used. Then water bath, iodine solution, and why we need a white tile. Now, the procedure for this, we might have carried out this experiment in the lab. It's supposed to be done in the lab. What we do, you pluck a plant from a leaf that has been exposed to sun for at least 2 hours. Then you get this plant and then boil it. So that you get someone mute. We have a new person who has come in. Oh. Okay. So I was saying that pluck a leaf from a plant that has been exposed to the sun for at least 2 hours, boil the leaf in water for 5 minutes. The reason for boiling this leaf in water 5 minutes is to burst the starch grain and make its cells permeable. Because remember, we are going to be testing for starch. We are going to be testing for starch in this what? We're going to be testing for starch in this leaf. We're going to be testing for starch in this leaf, and therefore we need to boil it in water. And what we mean by boiling, you leave it when that boiling starts. That's when you start counting the 5 minutes. So that is the reason I'll ask you at the end why we need to do that fast boiling. Now, after which we transfer the leaf in a beaker which contains alcohol. Now, the alcohol we're talking about is the other ethanol, which we called methylated spirit. And this kind of ethanol, ethanol is volatile, can burn easily. So we don't, after boiling the leaf in hot water, need to boil it in ethanol, but we boil the leaf by influx. By influx, we mean you get the ethanol put in a boiling tube or test tube, and then you boil it over hot water. If we look at the apparatus setup, unfortunately, I think it is not here. But if you look at your books, maybe you might have seen that arrangement where the leaf is boiled in ethanol, but the ethanol is not put directly on fire. That's what meant by influx, or warm it over a water bath for a few minutes. Okay, so it's the water you boil direct, then the steam from the boiling water is what boils the ethanol, because it's volatile. If you boil directly, it can catch what? It can catch fire. So you observe that the leaf turns white and the alcohol goes green. That means all the chlorophyll has been what? Has been removed. Now, after boiling and the leaf in the alcohol, you need to dip it in water again, because after washing it in alcohol, it becomes like very hard. It becomes very stiff and it be very brittle. Something brittle can break easily. So we need now to make it soft. How? By now dipping it in hot water for 1 minute. That's the reason why we are bring back this leaf into water for a second time. Now this time we leave it there. We dip it in hot water so as to make it what? Soft, and wash off the other alcohol, but mostly to soften it. Then after which, so you press the leaf in a dilute solution of iodine, and the observation, the leaf will turn black. And the leaf turning black shows that this leaf contains starch. Now, if you used a leaf which did not have starch, that is a leaf that you have distarched. Can someone tell me how we can distarch a leaf? Participants, how best can we distarch a leaf? Do you understand the word distarching? Distarching? Do you understand the word distarching? Can I get some response?
No,
Peter? Anyone who understands the
me,
meaning
I
of the
don't.
word? Yeah, I guess you don't understand, eh? Yeah, yes. I tell
Yeah.
823. You tell us your name and then give us the answer. Yes, please. Someone had their hand up. Is ITEL. ITEL A23. Who's okay? The person has gone off. Okay. Anyone who can try? How would we discharge a leaf or a plant? How do we discharge a plant? Yes, Rooney. Your hand is up. Rooney? Rooney? Yes, Rooney? Okay, anyone else who can help me understand what do we mean by how would we discharge a plant? How do we discharge a plant? Discharging is removing starch from a plant. If I want to remove starch from a leaf or a plant, what do I do to that plant? That is the question. Agaba? Hassan? Yes, Deborah. Peter? Okay, now, okay, Treasure? Okay, the way we discharge a plant or a leaf is by keeping it in darkness for a period of 24 hours. So you keep it in darkness for 24 hours. That is how we discharge a plant. Normally we use potted plants. That means you can move it from the sun and keep it where it has no access to light, and for a period of 24. After that period, all of the starch will be used up by the plant, and by the time you get that leaf, it will not have starch. If it is on the plant, maybe you can find a way of covering it with a black acavera, and so that is not exposed to light for at least 24 hours. That plant will be, that plant or leaf will be discharged. So that is how we discharge a plant, okay? Keeping it in darkness, a plant or a leaf, or keeping off light for at least 24 hours. All right. So if we use the discharged plant, a leaf from a discharged plant, it means they shall do the same procedure, but the observation will be different. Okay. This time when we have to test for starch, we shall not get the leaf turning black. What will it turn? Can someone tell me? What will it turn to in that case? If it doesn't go black, and that means shall be getting a negative test, what do we expect in case of a negative test? That is if starch is absent in that leaf. What do we expect? All right. So if it's not having starch, it has been discharged and used that leaf, then it will go into yellow, because it will turn yellow or brown. Yeah, it will take up the color of iodine. That is brown color. That means that starch will be absent in that leaf. So that is the experiment we can do to show that the experiment to test for starch in a leaf. I have a few questions before we move on to the next experiment. Can I have a response? First question is, how do we discharge a plant or a leaf? Question 1, how do we discharge a plant or a leaf? Anyone on? Give me the answer.
By leaving it in the dark for 24 hours.
Very good, okay? That is one. Now, second question. When you're testing for starch in a leaf, we boil the leaf in hot water for 5 minutes. Can someone tell me the reason why we boil that leaf in hot water for 5 minutes? A different person this time around. Can you please participate in this lesson? It is question time, please.
Okay, teacher, I'm trying.
All right. Name.
Yeah, to test whether it has chlorophyll.
Two.
To test the plant whether it had chlorophyll, the leaf.
The leaf, whether it has chlorophyll. Thank you for trying.
Yes. Okay.
Thank you for trying. However, we have been specific. The whole experiment is about that. Okay. But this is part of a procedure. Okay.
Okay.
This is part of a procedure, and the procedure was to boil the leaf in hot water first of all. There must be a reason for boiling the leaf in hot water. Do you have it in the notes? Okay, someone else to try again. Bridget.
Yes.
This
Bust the
Very good, eh? To bust the starch grains and make the leaf permeable, or to make the cells permeable, okay? Because remember at the end we add the iodine, okay? So the iodine must be able to penetrate, all right? So this is the reason why we boil the leaf in hot water first time. All right, now second time, the second procedure, we get the leaf and then we boil it in methylated what? We boil it in methylated spirit or alcohol, all right? And why do we boil the leaf in methylated or spirit or in alcohol? Anne, can you give us the answer? If you can hear me, Anne. Okay, Charles Agaba, can you hear me? All right, anyone? We're running out of time. Anyone can give me the answer? Do you people know how to use this feature of raising up the hand? Yeah? Hello?
Yes, teacher.
I have asked a question.
Okay.
And I expect a response. The question was,
Yes.
why do we boil the leaf in spirit? Question written there. Answer. Any participant to give me the answer? Okay. Now we say the reason why we boil the leaf in ethanol is to remove what? Chlorophyll. That's the reason why we boil the leaf in ethanol. Okay. Now, next question. After the boiling the leaf in ethanol, we say it becomes brittle. Okay. What can we do with this brittleness? Because we need it soft. When this brittle means can break easily, what do we do in this case? After boiling the leaf in ethanol, we say it becomes brittle. Then what do we do with this brittleness? How do we go about it? Answer, please.
We boil it.
Dipping
again
it in
warm water. Hot warm.
Very good. Okay. We dip it in
Yeah.
hot warm water for one minute. Okay. And when you do that,
Yeah.
it becomes soft. So simply say it is to soften the leaf. Okay. The reason is to make the leaf soft for the next procedure. Remember, we are testing this leaf to whether it has starch. We are testing for starch in the leaf. So that's the reason we dip in hot water for the second time. Then the second question: how do we confirm? How do we confirm that starch was present in the leaves? How do we confirm that starch was present in the leaf?
The leaf turns white.
Another trial.
The leaf turns black when iodine.
Yeah. Very good. Eh? So we'll get this leaf, place it on a white tile, okay? And the reason we press on a white tile, we want that background, the white background. Then we get an iodine, and then we apply iodine what? Solution. When we apply iodine solution, then the leaf turns what? The leaf turns black to indicate that starch was present in that leaf altogether. So that has been the experiment to show or to test that starch is present in a leaf. Briefly, before the end of our time, let us talk about the next experiment. The next experiment, can someone read us the title for the next experiment? Look at the shared screen.
Experiment to show that oxygen is given off during photosynthesis.
Very good. Now, when we are looking at some of these experiments, and from our understanding of photosynthesis, when it takes place, there is oxygen which is produced. Now, we want to see if really it is true that during photosynthesis, oxygen is given off. The materials we need, okay? Who's reading us the materials needed for this experiment to show that oxygen is given off during photosynthesis? Someone read us the materials. Okay.
Water,
Very
beaker,
good. Okay.
test tube,
Very good.
fresh beads, in brackets, elodea,
Okay.
Then sodium hydrogen.
Then sodium hydrogen what? Carbonate. Now, let's look at the setup. The setup of the experiment, all right? Maybe we shall understand better. Okay? Can you clearly see the setup on the screen? Yes, we can see it now. So what we see is the Elodea plant. This Elodea plant has been enclosed in a funnel which is turned upside down, and the funnel is standing on a wooden block. Then we have water, which water is dissolved with sodium hydrogen carbonate, which is the reason for that. Then we have a beaker which is turned over the funnel, that short stem of the funnel, and then we see bubbles of a gas, and then some space for collecting that gas. Shall later know which this gas is. Now, that is the setup from the whatever the setup we have, and we say we get a fresh pondweed. For example, we can get there. This is a water plant. Okay, which is put in a beak of water with dissolved sodium hydrogen carbonate. Now, the role of sodium hydrogen carbonate is to provide carbon dioxide. Remember, this is a water plant, and we need carbon dioxide for photosynthesis, okay? So being an aquatic plant, we have to make sure that the water we use has what? Enough carbon dioxide. Then this plant is covered with a funnel, which is inverted with a test tube with water. Which is inverted with a test tube. And then it is supported by wooden blocks. I've talked about those blocks in the setup, so that this funnel can sit on those blocks. Now the setup must be left for a period of 6 hours. And whenever you're carrying out an experiment, you must define the time of an experiment. If you describe an experiment without giving the time for an experiment, then you lose the mark for the results or the observation, because it means that you cannot get those observations since you have not talked about time. So it is very important to know how long the experiment should run. So this time you're leaving it for a period of 6 hours. Then two, when you are describing an experiment, you have to talk about a control, what we call a control experiment. The control experiment, in this case, we have to get another plan, okay? A control experiment where we don't place the same setup in light, but we leave it in what? In darkness, okay? Remember, we want to show that light is given off during photosynthesis. Therefore, that's why we want to prevent photosynthesis by keeping the plant in darkness for a particular period of what? Time. So we have two setups, the real experiment and then the control experiment. For the control experiment, we leave the setup in darkness, okay? And later, of course, if we look at the setup again, you see, if we leave it in light for some time, we see bubbles of this gas being produced, and the bubbles of the gas are collected in the space within the test tube, which we inverted over the funnel. And what we see here is observation. We get this gas which has been collected, and we test the gas. We test it whether that gas is oxygen. We don't know how we test for oxygen in the laboratory. Okay, the lab test for oxygen, as we learned it in chemistry, we use a glowing spring. Okay, and we know oxygen lights a glowing spring. So once this gas is exposed to a glowing spring and is lighted, we confirm there is no, the gas was oxygen, and this gas was produced by the photosynthesizing what? Earthia plant. I'll ask one of you to tell me the expected observation for the other control experiment. Which was kept under darkness. Yes, Marcus. Sekide. Yes, Sekide. Can you give us a response? Yes, Sekide. Yes, Sekide. Yes. Sekide. Okay. Anyone to give me the answer to my question? The question was, I've given you the observation for this real experiment, where the gas collected lighted a glowing spring, which we confirm it is oxygen gas produced during what? Photosynthesis of their plant. Now, what would we expect if the setup was kept in darkness as our control experiment? Time check is 4 minutes past 3. We should be summing up this lesson. Yes, Deborah. Deborah? 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 Okay. Anne? Kaitesi, are you still there? Katrina? Darius? Peter Clever, you please speak to me. Fiona Segawa-Haman. Segawa-Haman. Mark Trevor. Deborah, then mute if you're not talking to me. Yes, Katrina. Angela?
Yes,
Yes, please.
I have no answer for
Okay. All right. Now that you understand that we have kept this leaf in darkness, of course, we shall not collect the gas, or whatever gas will be present there. When we test it with a glowing spring, it will not be relighted, which means it shows that no oxygen will be produced. So we shall confirm that really oxygen is produced only during photosynthesis, when the plant is exposed to what? Sunlight. The fact that the one kept under darkness is to want to confirm our results that you not get oxygen gas being released. Okay? Or whatever gas, if you find any gas in that test tube and test with the glowing spring will not be relighted, meaning with no oxygen. So we confirm that oxygen is produced. Okay? Oxygen is produced, it's given off by plants only during what? Photosynthesis. How together? Can I get any question from any of you? Should we continue with the other experiment? Or leave it for tomorrow? Okay. We really appreciate the time you have spent with us. However, what is still lacking is communication. I need your feedback. When I ask a question, I expect you to unmute and then give an answer. It can really give us a good feedback, a good understanding of what is taking place. Okay. For today, we have had a recap of what we did at school about nutrition and living organisms. We have looked at briefly the types of nutrition. Just a minute. Yes. We looked at the types of nutrition, heterotrophic, autotrophic nutrition. Looked at the heterotrophic organisms and how they can be grouped: saprophytic nutrition, some parasites, holozoic feeders, insectivorous, and epiphytes. Then we didn't look much at the saprophytes, but we know sapro means rotten, phyto means plants. Okay, so the saprophytes obtain food from dead and decaying organic matter. They include like molds, mushrooms, and many bacteria. Then we also looked at things like what we expect from food. We expect energy from food. We expect food to be protective, like vitamins and minerals. We expect food to provide energy. We expect food to build the body, like the milk, meat, the proteins do that. Then we looked at the photosynthesis as a term. We defined it, like we did it in class. Then we looked at the products and then the raw materials, the reactants, which is carbon dioxide and water, the raw materials, okay? Then the requirements for photosynthesis which we looked at, that was chlorophyll, carbon dioxide, then water. Then we looked at a little about the limiting factors, that was light, temperature, and carbon dioxide. Then also at other factors which affect the rate of other factors which affect the rate of what? photosynthesis. Okay, and the come back with the balance. Okay, other factors which affect the rate of photosynthesis, and then we mentioned those factors like oxygen. I told you how too much oxygen can destroy the photosynthetic tissue, and then mineral salts, pollutants like ozone, sulfur dioxide. We saw how they can affect photosynthesis. Then chlorophyll content, then accumulation of the carbohydrates. If you allow them to accumulate and they are not removed, the photosynthesis will reduce. Then the number of stomata in a leaf, then the surface area of the leaf. Then finally, we have looked at the experiment on photosynthesis. One experiment we have looked at was to test for starch in a leaf, and we said at the end we need a plant that has been in the sun for some time, and that plant we get a leaf from it. When we get that leaf from the plant, we test it for leaf by boiling it in water for 5 minutes. We have said the reason to burst starch grains, after which we boil it in ethanol, reason to remove chlorophyll, after which we dip it in water because it has become hard and brittle, so we need to soften it by soap. After that, we press it on white tile. After pressing it on white tile, we now add the iodine, and when it turns black, it's an indicator that starch was present. The second experiment we looked at was to show that oxygen is given up during photosynthesis, and this time we have used the water plant out there, which we have placed in the water and covered with a funnel. Then we collected the gas, which was produced during the process of photosynthesis, because the plant was exposed to light for 6 hours. And the gas was collected, which we tested with a growing sprint, and realized that the gas